use your voice to let me know. Okay. Okay, good afternoon, everyone. We will talk one of the topics in the last unit, fourth unit, and it is about the use of humanized or humanized mice models. So if you want to generate humanized mice, we will discuss what they are and then how stem cells are important in biomedical research in this view. So if biomedical research is about studying the diseases and trying to come up with new ways of treating diseases or preventing, managing, and treating diseases. And for that, we usually rely on several model organisms, like zebrafish, that's one common one. Drosophila is another common one. C. elegans is another. And we can use any of these, but whether is it relevant or not is one question. And we are so diverse in these. So in terms of our evolutionary lineages, what we are closest is mouse that we use, mice and rats, because they both are mammals, right? So they're likely to be very similar. But then there are also a lot of differences between humans and mice. So while we share a lot of similarities, there are also prominent differences, because that is how the diseases also are different. So we typically, I hope you know about HeLa cells, the cancer cells that have been isolated from breast of Henrietta Lacks. That is a huge controversy also involved with that. But you can see for almost 60, 50 years, we have been using these cells for biomedical research. Of course, there are many other cell lines as well. And we use it for almost everything, including research in space as well, how the cells will respond to growth in space, outer space. So when we use these cells for studying effectivity of drugs, one of the problems is that the cancer cells, for example, they undergo mutations, further mutations with number of cell divisions. And then what is grown in one lab, and when it passed on in different labs, different mutations occur, and they all have different differences in the way they respond to to drugs. So we cannot really say what is proven in cells we can translate it to humans. That's not that is one of the complications that we have because of this chromosomal instability. So how else can we study these humans? The problem is we cannot try everything that we get on humans directly, because it's going to, if there is any effect, we are going to cause more harm to the individual than actual benefits. So we should have tried these drugs on other systems, organs or organisms or tissue cells, and ensure that they do not have any drastic or lethal effects. Only then we can use it as a medicine, or we can try on humans. At least that way we are sure that there are no known effects or known ill effects on the human. So I hope you remember studying about this, the biology for engineers, I think. You have learned about organ and chip, where you can use different systems like this. And each of them, you can have different cells, and then they are connected through small, fine tubes or tunnels. And they kind of function as if it is a miniaturized artificial organ. And then we can probably also test different drugs on or build disease models on this chip. While this is still underway, we cannot, we have to still look at, we need more and better ways of, more accurate ways, so that we can try out more drugs or screen more drugs for effectivity. So we have to, even now, we rely on mice, although there are ethical issues. Maybe PETA and others will have concerns about using animals models for testing drugs. Testing drugs is different from testing cosmetics, isn't it? Cosmetics is for luxuries. I'm certainly against those things, but if we want to develop drugs for the human diseases, something has to. There are going to be ethical issues, of course, because we are going to try it on organisms. Mice are favorites because they grow, their lifespan is small, and they grow, come to maturity very fast, and then they give birth as well. The generation time is short. So we can use mice, but they are mice, right? They are not humans. We cannot necessarily translate anything that we find true in mouse models to that of humans. They may be, most likely, but there will also be huge differences. Moreover, say diseases like some tuberculosis and others, or HIV, for example, they happen on humans. They don't necessarily, they cannot make that, those, the virus cannot infect mouse cells. So we cannot build disease models in that sense. So we need to do something about these mice, and that is called humanization, in the sense that we are going to put in some human genes or cells, tissues, or organs. And if you make a live mouse like that, then it is called as a human mouse. So a mouse that carries human cells, genetic information, tissues, they are referred to as humanized mice. So this is an important step, because we want to have these, we want to, before we try it onto humans, we want to test that it works. And so we are making humanized mice, we are generating a preclinical model. And once we know that there are no ill effects, and probably there are desired effects, then we can probably try it on real clinical trials on humans, right? And probably also onto the patients as well. So that's why humanized mice is kind of translator in a way between preclinical to the clinical side. I hope you remember what we will discuss in this lesson is we will see if there are, how human genes can be put in, and we will also see how different cells and tissues are put in. This is what the focus is. I hope you remember from transgenic animals. I hope you watched the videos, and there are two different methods: direct microinjection method and engineered stem cell method, right? This is used to insert genes, delete genes. You can also have knockdown of genes. And in that, we also learned about how to generate the mouse, right? In that, we take a superovulated donor and a normal male. We take their gametes, and in vitro fertilization would have occurred. And to that, we add DNA directly. This is the microinjection method, direct DNA injection method. And then we will transfer the embryo or the manipulated embryo into the pseudo-pregnant female. What is pseudo-pregnant? Which would mean that the recipient female is allowed to mate with a vasectomized male. So physiologically, it will develop into, or will be prepared to conceive and hold on to the eggs that we are going to put into the uterus of this female. So it is a pseudo-pregnant one. And then we would get the transgenic powers. If it were engineered embryonic stem cell method, we would have taken the ICM, collected cultured the embryonic stem cells. From there, we would have transferred the gene of interest, we would have done positive selection, negative selection, and then we would have gotten the engineered embryonic stem cells. From here, we are required to develop a full organism. Here, these are only cells, and they are pluripotent. They cannot give rise to complete organism. So what happens is we take these cells and we put it into a recipient blastocyst. And from this blastocyst, we will implant this blastocyst into a pseudopregnant female, and we would get a chimera, right? Because there are two different ICM here. So we will get a chimera. From the chimera, we would get a first transgenic animal like here. So when we put, say, here are the engineered embryonic stem cells, we put it into a blastocyst. This is a surrogate mother, and we got a chimera. When this chimera is allowed to mate with another one, it will generate pups, and one of them or some of them would be the transgenic ones, right? So what we are going to do now is understand how to put the genes into mice. Okay, these are some of the popular methods. One of it is this first one is humanization by using YAC or BAC. YAC is yeast artificial chromosome, or BAC is for bacterial artificial chromosome. And this is an additive transgenesis, which means you're not deleting anything. You're putting in an extra copy of human sequence. You're not deleting anything that is present in the mouse. Instead, you're adding, say, for example, here, CDEFG sequence into it. So you can do it by direct injection of the DNA into the zygote, fertilized egg, and then put it into surrogate mother, and then check on the babies using molecular techniques like PCR, southern blotting, and others. And then you can figure out which of these founders have the DNA integration, and then you will get you'll find out the you would have gotten the humanized mouse. You can also do it by fusing a yeast cell containing YAC, that is yeast artificial chromosome, and the similar things go through it. The other ways are using by homologous recombination. In this case, what we are doing in the process, first one is additive transgenesis. We have just added here, and then we are allowing it to mate with a knockout mouse, so that one of them or some of them will get, we will have to identify the organisms that have only the human genes and not that of the homologs from the mouse. Okay. The next one is using homologous recombination. In the previous one, the integration of the genes could be anywhere, randomly integrated, unless we are targeting it. Here we want to use homologous recombination to eliminate, to replace the mouse genes, mouse homologs, with that of human homologs. So here we have CDEFG, and this is of the human genome. Okay, and here we are having the embryonic stem cells of the mouse, CDEFG. So if we put in a vector like this with bacterial artificial chromosome and allow homologous recombination to happen, AB is there, here to AB, FHF, here and here, there could be homologous recombination. Because of homologous recombination, what may happen is the loss of the replacement of, or these are removed, and you will get CDEFG from humans. Okay. And I hope you remember these are site-specific recombination. And then when there is recombination between these two, this part which we have added for positive and negative selection can be removed. So now we get EFG, CDEFG of a mouse with human CDEFG genes. So we can also do it by another replacement. Here, the replacement is by using site-specific recombination. Here, we are using homologous recombination for between these regions so that we'll get the integration of the human genes. In this case, we are adding site-specific recombination sites here and here, right? And the same things, we should have first engineered site-specific recombinations into the mouse chromosomes like this and here. Okay, this is another, there is a prior step of how you would have constructed this mouse embryonic stem cell chromosome with pre-integrated site-specific recombination sites. And now we take the human recombination, human genes, C, D, E, F, G, along with these restriction sites with the arrowheads shown here, and allow these sites to recombine. So a cut will be made here, and a cut will be made here, and similarly here and here. And then this whole DNA will be moved to this place. And now we have integration of CDEFG into the site where there were CDEFG, mouse CDEFG, right? So these are ways of how to replace the mice genes and incorporate human genes. There is also one more step called as using HAC. HAC is for human artificial chromosome. I mean, basically, yeast artificial chromosome and human artificial chromosome are very similar. They are linear and they have telomeres on their ends. They have centromere, which is, and they have also the autonomous replicating systems. That is the origin of replication. Telomeres are responsible for stability of the chromosomes, solving the end replication problem, prevent recombination with other ends of the chromosomes, end joining with other chromosomes. And we need origin of replication for every replicon. I mean, that only if a DNA is able to replicate, and then it is genetic information, or it should be associated with that. If there is no replication, the DNA is not going to be inherited by anything else. The third important point I said was it should have a centromere. And the centromere is responsible for segregation during cell division, mitosis as well as meiosis. So we can construct human artificial chromosomes, and you can make all the genes that are responsible you want to transfer locate, like A, B, C, D, E, F, G here, and then make a microcell and then fuse it with embryonic stem cell. So we can deliver this DNA, this chromosome, into the mouse, a mouse embryonic stem cell. Okay, and then it is kind of, it will keep on. It can be as an, it will be maintained as if it is an extra chromosomal element within the body of the mouse. Okay, we just added a completely new chromosome. Why is this better than directly adding randomly? This does not require this random integration is always a problem, because we don't know which other genes are affected because of the integration. In this case, we are adding a new chromosome itself. So we are not altering, or we are not disturbing any other genes in their locus. Okay. So that way we can produce mice that have human genes, because of which they will have human proteins. And then the interactions between, say, for example, pathogen like HIV can happen. So there is one more problem. If we want to put in cells, for example, human cells into mice, we want to put human tumor into a mouse and try out a drug. The problem is that the mouse immune system would actually eliminate, graft rejection would have occurred. We learned about this. We discussed this before. So what we can do is, there is a disease we learned about, SCID. SCID mice, which is severe combined immunodeficiency. Sometimes there will be mutations within the genes involved in immune system, because of which they have decreased immune response and they cannot fight infections. So typically, it may also occur in humans. This person has that SCID, and because of which the person has to live forever in that bubbles, unless they are provided with other means, with technology, probably. So we have to first, if we want to put in cells, if we want to put in human cells or tissues into mice, we need good models which do not have immune system or active immune system. So in mice too, they found some of them have SCID, that is severely combined immune disease. And there are also other forms like NOD, that is the non-obese diabetic. It also has some immune deficiencies. So they made a cross between them and generated mice with NOD as well as NOD and SCID. That means they are severely deficient in immune system, natural killer cell activity, and decreased innate immunity. So some people, by using genetic manipulations, they have been able to delete interleukins and other things as well from these organisms. So from this NOD SCID. So these are all different strains of mice in which different types of genes were deleted. Okay, all majority of them are associated or involved in immune responses. So this is basically the base, in a way, onto which we can probably include the human cells, and then probably develop disease models and screen for drugs that are effective, or screen for strategies that are effective. Okay. So there is one more point that we need to put in. And now, when we are putting in human cells, we call them, they put it as HU to denote that these are humanized mice. Okay. So different tissue grafts can be put in different locations. Like we discussed when we were talking about cancer stem cells, we discussed that some of the areas where subcutaneous, intravenous, we learned about different sites of injection or transplantation for these biopsy samples. Similarly, there are several types of tissues, say fetal and thymus fragments, you can put under the renal capsule. So PBL, that is peripheral blood lymphocytes, they can be put in spleen, etc. Okay, these are in the face vein also is used. Intravenous tail vein is also used sometimes. So it depends on what tissue, and then they place these cells in that particular body. And now the important point anyway is that the mouse that we choose is the one that doesn't have very effective immune system, or its immune system is very, very weak. So the cells that we are putting in, they will be able to survive in those respective areas well and function as if it is in the human tissue. So here we'll try to see how these humanized mice can actually be useful. This is NSG mouse, which is not SCID and gamma deficient, or this is severely deficient with immune system. So they do irradiation of the bone marrow. They remove the bone marrow and irradiation, and after 5 weeks they will inject human hematopoietic stem cells from humans. And after about 16 weeks, the human hematopoietic stem cells will start developing several cells from the humans, and now we have a humanized NSG. Now we can put in the tumor samples into the mouse and allow the tumor growth and see how, and we can then also use antibodies, monoclonal antibodies. We can try different types of monoclonal antibodies or drugs and see which one is effective in these mice. And apparently, using these monoclonal antibodies, it is showing better results. So you can probably translate that into human subjects, and eventually we can develop a treatment strategy or approach for cancers. Okay, that is how these mice are used. We can do many other things. We just discussed a little bit of this stuff. Here is another one about how to validate. Okay, these are, I'm putting it here so that you will have your own ideas in the future. You can take the patient cord blood and expand them and then put it in and graft them into mice. The mice, of course, are skid at something like NSG, and see the assess humanization. That means we should have taken the cells and checked how many of them are, what percentage of the blood contains human tissues. We should also take the biopsy from the tumor and put it into the mouse. So we have human tumor, we have human blood, human immune system to an extent. And now see how the progression of the tumor is happening. So we are trying to see what is happening with the human because of the biopsies and also after the therapies. We can also try the same things as we are doing with the human, or try alternate therapies to see what is happening to these tumors. If it is getting killed here, we can probably try the similar strategy here. Or when we do parallelly, we are trying this and same thing here, we'll get a validation of how good is the humanized mice concept to start with. And I think there is considerable success in this line as well. So we can try many things. It's not just about cancer itself. Here is a where is it? Yes, here is a mouse model for treating with or trying to find cures against HIV. Take mouse, humanized mice, and then we can use the one second. I wanted to have, yes, we can use anti-HIV genes modified cells. Okay, we have, say for example, CD44 is through which the HIV enters into the cell and then destroys those lymphocytes. Then we want to have cells that are resistant to HIV entry, right? So we should have taken those cells that are probably somehow we do by other genetic manipulations. We put in those cells into the mouse. Now, this mouse is having the cells that are modified, and then we give the HIV challenge. That means we are injecting HIV. And then we are checking if the cells that we have put in are getting infected or not, right? That you can see if they have some degree of immunity or not. HIV, what does it do? It infects the immune cells, because of which the person becomes it is acquired immunodeficiency syndrome, right? It is a plethora of deficiencies in which there is no immune system, because of which even minor small infections also might become severe. And we can have different types of strategies, and we could study them in the mouse itself. So I will share this, and I would want that you try out different. You try to read. There is also a table that I have added here. Each of them with different, a little bit slight variations. Okay. So I hope you understood the humanization of mice. We are concerned about it, and we should be able to see how important it is. Okay, first thing about genes, how to put that is with recombinant DNA technology, or you can call it genetic engineering, somehow putting in the genes, replacing the genes of mice with that of humans. Not all genes can be just replaced like that. Some may work, some may not work. And if they don't work, then we better try to put in human cells into them and then try out, build disease models. We can evaluate the how drugs work before we try it on any of the humans. If they are not good, if there are any serious side effects, we wouldn't have tried those on the humans. That's why humanized mice are very important for research and also in preclinical studies. Okay, I think I will stop here. If you have any questions, you can try. And these PPTs are already shared with you. So I would encourage you all to have a look at that before. So any other questions? Any questions at all?
No, sir.